So apart from being one of the great disasters of human history, the 1914-18 war marked the beginning of an end to when European civilization dominated the planet. It was a great divide, I think. While Australia was a sovereign nation in 1901, the national mindset was very much embedded in the idea of British exceptionalism and the British Empire. Australians joined the first Australian Imperial Force, the AIF, in enormous numbers, an enthusiasm that was, by the time World War II kicked off in 1939, very much dimmed, until Japan's southern thrust made it obvious that Australia was at risk. And a lot of people then joined up for the second AIF. At least in the early days of World War I, before the mass slaughter really got underway, there was a kind of romantic thing about it, an element of romantic patriotism for what seemed initially to be a great and noble adventure. War poet Rupert Brooke, for instance, the soldier, "If I should die, think only this of me, that there's some corner of a foreign field that is forever England, there shall be in that rich earth a richer dust concealed, a dust whom England bore, shaped, made aware." A fine poet. Commissioned in the Royal Navy Volunteer Reserve as a sub-lieutenant, the 28-year-old Brooke sailed as part of the disastrous Dardanelles campaign and died aboard a French hospital ship from a mosquito bite that turned septic. He lies in a corner of an olive grove on the Greek island of Skyros, a sad waste of a bright spirit who is described by W.B. Yeats as the handsomest young man in England. I always wonder at those statements, you know, the cleverest man of his year and so forth. I don't know what sort of survey techniques they use, but so many were to follow Brooke. Some 37 million soldiers and civilians died in 1914-18, and Brooke experienced a much calmer passing than was the case for many of our Anzacs. Unlike Simpson of Donkey fame, most were not English born, though many still talked of England as home and were embedded in the idea of empire. Up until World War I, Brooke's fate to die of infection rather than wounds was the norm for soldiers. Apart from being the Gallipoli centenary, 2015 also marked the end of the American Civil War, where repeating rifles, revolvers and machine guns were used for the first time. In that conflict, which comes just at the time Pasteur was establishing the German theory of infectious disease, and Semmelweis and Lister were initiating the linked practices of medical hygiene and antiseptic surgery, deaths from infection versus combat were roughly 4:1. 4 times as many people died of infection as of being shot. At Gallipoli, that 4:1 ratio had dropped below 1:2. 5,480 were killed in action, 2,012 succumbed from their wounds, a lot of that was likely due to infection, and 665 died of disease. Though this was only about 50 years after Pasteur's experiments that formally established the germ theory of infection, and 60 years after John Snow showed that typhoid was spread from contaminated water pumps, the basic principles of hygiene, along with the necessity for clean water, were already well established, understood and applied. At least some British soldiers were immunised against cholera, and Australian troops were given typhoid vaccines on the voyage across to Egypt. In addition, both the intensity of the fighting and the dry conditions could have led to better outcomes than those experienced in the 1861-65 Civil War. And there were major advances in surgery and nursing, including the infusion of warm saline, which also helped to revive many patients who were very cold when they came in from the battlefield. And we've all heard stories about people who were just classed as being dead when they came in from the battlefield and put aside and then found to be alive. And the nurses who were doing the initial triage at the time said it was often extremely hard to tell whether someone actually was alive. They were so cold. Despite Landsteiner's discovery of the blood groups in 1900, the transfusion of citrated blood did not get underway until 1917, when it was rapidly expanded with the arrival of the United States forces on the Western Front. While Paul Ehrlich had developed arsenophen, or salvarsan, an arsenical, the first magic bullet for the treatment of any infection, syphilis, in 1909, the idea of treating infections with drugs did not progress much through World War I. The situation had greatly improved when 20 years later the world embarked on World War II. By the mid-1930s we had the sulfonamides, first Bayer's Pronosil and then sulfanilamide. In World War II, US combat troops were issued with sulphur and told to sprinkle out on any open wound, while after being released from internment, Australian-born Victor Chakotjas with C.K. Hughes synthesised sulphur guanidine that prevented the deaths of many of our soldiers fighting in the tropics from dysentery. Discovered by Alexander Fleming, Oxford-based Australian Howard Florey, and Austrian emigre Ernest Chain pioneered the development of penicillin. Scaled up for massive production in the USA, penicillin was available by D-Day in 1944, with the consequence that deaths from infection versus combat were down to 1:9 for US troops at that time. On the World War I Western Front, the soldiers of the first AIF were to suffer along with everyone else the constancy of scabies, lice infestation, and the horrors of trench foot, which could lead to amputation and was best dealt with by having a supply of dry socks and smearing the feet with whale grease. Whale grease also protected fighter pilots from the blowback from machine guns, which were firing straight in front of their face. It didn't protect them though from the castor oil that was used in the fuel, and that gave a lot of them nasopharyngeal cancer later. Wounds might be treated with iodine, carbolic acid, hydrogen peroxide, perchloride of mercury, sodium hypochlorite, boracic acid, salicylic acid, chloride of zinc or potassium manganate, either alone or in combination in liquid or paste form. An example is being bipped by smearing with bismuth, iodine, iodoform paraffin paste. Post-surgery, large open wounds were irrigated at multiple points, up to 5, with Carrel-Dakin solution, hypochlorite. Carrel, of course, is the guy who developed tissue culture. Of course, none of this was of any value whatsoever for treating respiratory infections. Then there was the 5-day French fever caused by Rickettsia quintana, while thousands died of tetanus and gangrene. And the nursing sisters and the VADs who cared for them suffered from severe infections, especially to their hands, from the suppurating wounds they tended, while they also caught the endemic diseases of the trenches, typhus, dysentery, mumps, and later influenza. Antitoxins, or serum against tetanus and diphtheria toxin had been available since the early 1900s. And fighting in the manure-laden fields of France and Belgium, the Germans in particular developed prophylactic serum dosing as a preventive against tetanus. Though they ran the risk of anaphylaxis, the results were generally beneficial. As the war progressed, wounded soldiers might first be assessed at field dressing station, then passed on to a casualty clearing station, where they would be given anti-tetanus serum and perhaps have their first encounter with a surgical team prior to transfer to a large base hospital. Their likelihood of survival was greatly increased if they made it that far. But one of the longer-term possibilities was the development of gas gangrene caused by Clostridium perfringens. The French produced an anti-serum that proved to be of little value, and apart from amputation, the most successful treatment for gangrene was debridement to remove necrotic tissue and drainage. In World War II, prisoner of war Dr. Rolly Richards, lacking the most basic drugs, used essentially the same approach to deal with the horrific tropical ulcers that afflicted captive slave labourers on the Burma-Thai Railroad. If an organism could be seen by the high-power lens of a compound like microscope, like malaria, or the etiological agent could be cultured in or on various liquid or semi-solid media, we had by 1914 a reasonably good understanding of some parasitic and bacterial diseases. Due to the availability of readily manipulated experimental systems, we also knew by the end of the 19th century that some infections could be transmitted by an agent or infectious fluid that would pass through ceramic filters with pore sizes small enough to remove any known bacteria. These things have been in use for a long time to clear up water supplies. Furthermore, these viruses a term coined by the Dutch microbiologist Martinez Beyering, could not be cultivated in any known bacterial culture system, though they might, for example, grow on the leaves of a living tobacco plant. Tobacco mosaic virus was in fact enormously important in the early history of virology, with transmission studies soon suggesting that human yellow fever and bovine foot and mouth disease were also virus infections. In addition, Peyton Rouse, who ran a blood transfusion service on the Western Front in 1917-18, had shown in 1911 that a cancer of chickens, Rouse sarcoma, could be transmitted by a cell-free filtrate. There we actually used lab-grade filter paper for the experiment. Back through history, smallpox virus had been transmitted from person to person by the practice of variolation, where scab or pustule material was scratched into the arms of children or blown up their nose to give a mild case of the disease that provided long-term protection. The same approach had been tried with the cattle disease rinderpest, but the practice proved of little value. Otherwise, the suspicion that any particular infection was caused by a virus would have rested on an understanding of the epidemiology, along with the failure to culture a causative agent. Intriguingly, though no other virus had been grown by other than animal transmission, we had a live vaccine against smallpox, vaccinia grown in calf skin, and the attenuated vaccine from spinal cord of infected rabbits for rabies, developed by Pasteur. And since the 1890s, Robert Koch, who had first cultured the tubercle bacillus in his disciples, had been trying to develop a vaccine against tuberculosis. The BCG vaccine was developed from 1905 and was first used in 1921, a value mainly in children that has been in limited use ever since. When it comes to respiratory infections, the main military concern was tuberculosis, which was prevalent throughout society. Radiological screening of recruits was possible, but the facilities were limited and it was not used in 1914. In the Army records, for instance, indicate that at least 10,000 of US Army records indicate at least 10,000 of those who fought in France had TB. There were no drugs, and those afflicted were considered to benefit from good food, sunlight and fresh air. These themes, of course, persist in the general population. When it comes to returned Australian soldiers, more than 3,000 later died of tuberculosis, and so many, with lungs irreversibly damaged by gas attacks, were particularly susceptible to various respiratory infections. When influenza hit the Western Front in 1918, neither the medicine nor the science was sufficiently advanced to be of any great help. One positive since the American Civil War some years back was the emergence of nursing services. In general, nurses were of considerably more value than doctors in the flu pandemic. And as told in Tom Keneally's novel, The Daughters of Mars, many nurses succumbed to the infection. Bacteriology was reasonably sophisticated, and workers in the US, for example, produced a killed combination vaccine incorporating several strains of pneumococcus plus Haemophilus influenzae, also known as Pfeiffer's bacillus. Perhaps this was of limited value in preventing deaths from secondary infection, but the available data is not sufficiently comprehensive to make a conclusion. Even when influenza virus was finally shown to be a reality in the 1930s, that was established by transmission studies using pigs by Richard Shope or humans and ferrets, Wilson, Smith, Andrews and Laidlaw. It was not until the 1930s before the egg inoculation techniques developed by Goodpasture and used so powerfully by Burnett as a research tool came on the research scene. Burnett made an egg-grown influenza vaccine during World War II, though the great fear that the conflict would bring on a repeat of 1918-19 did not materialise. As influenza researchers, we have of course lived with the consciousness that a terrible influenza pandemic is an ever-present reality. The risk seems magnified by rapid international air travel, though sometimes I wonder if more numerous infection events may not lead to enhanced background T-cell and antibody immunity. We have antivirals with more in development, though they have been They have to be used early in the infection if they are to be of benefit. For prophylaxis, particularly to protect frontline personnel, we could rapidly produce protective humanised monoclonal antibodies. Alternatively, if shown to be safe, gene transfer could be used so that individuals make their own cross-reactive immunoglobulins. Broad-spectrum antibiotics are available to limit the severity of secondary infections, and ECMO saves some lives, though some facilities would be, most those facilities would be rapidly overwhelmed in a major outbreak. Still, at the back of our minds, particularly for those who have seen a virulent avian influenza A strain destroy poultry populations, is the realisation that fatal influenza can be an extraordinarily acute disease. Then, at the acute phase, we also need a much better understanding of what should be done medically to limit cytokine shock. And how do we treat a patient who can't get enough oxygen to the late pulmonary compromise associated with tissue repair? With ever better technology and research tools like cytokine knockout mice, these processes are gradually being unravelled. And though vaccines have been variously great or not so great in different epidemics and pandemics, it's clear that we need much better products that can be produced at scale much more quickly. We've come an enormous distance since 1918, and that's what we're going to hear about at this meeting. But it's also the fact that there's still a way to go. So if you're a young influenza researcher, don't despair. There's plenty to do. Thank you.